You have helix, turn, helix. So if it is a, are termed as a super secondary structure, or they are called as a motifs. This is because in many proteins you will find the similar pattern. So it's a unique pattern of the secondary structure which has been referred as a super secondary structure. So if I find three different proteins having a helix, a turned helix, that means alpha helix, turn, and then alpha helix, we will call them as a motif, which is having helix turned helix motif. Is it clear? Okay. Now some of these arrangements are regular kind. This part we have already discussed. They maybe have alpha helix, beta sheet, and elements of secondary structure. Now coming to the tertiary structure. What is the tertiary structure? Tertiary structure refers to amino acid residues that are far apart in the sequence and to pattern of disulfide bonds. Now, whenever we have talked about the secondary structure, we have referred to some of the patterns or some of the arrangement, spatial arrangement, which is happening because of the vicinity or neighboring amino acid. But when we are talking about the tertiary structure, we are going to refer to spatial arrangement of amino acid residues that are far apart in the sequence. So suppose we are talking about the interaction about the fourth residue in a chain, or every fourth residue in chain, that means I'm talking about alpha helix where hydrogen bonding occurs. We are talking about the secondary structure. Suppose I am talking about interaction between fourth amino acid residue and 100th amino acid residue, which are far apart in the primary sequence, but because of their folding, because of their spatial arrangement, they have come close to each other, and that is what refers to the tertiary structure. And that's why you have seen tertiary structure is a quite folded structure. And then this tertiary structure also arises because of the disulfide bond, because these disulfide bond will be formed because some structure has come close to each other. Because of that, your disulfide group, they come close to each other. Sorry, close to each other, which leads to the disulfide bond formation. And thus, this disulfide bond formation keeps the tertiary structure in a particular fashion. So this part we have already discussed in a couple of lectures. Now we are going to discuss about the proteins that contain more than one polypeptide chain. So how do we find out whether it's a one polypeptide chain or more than one polypeptide chain? Can anybody tell how to differentiate between one polypeptide chain and more than one polypeptide chain? So far, up to tertiary structure, we have discussed that this interaction is within the one polypeptide chain. So how you recognize? Any one of you? One polypeptide chain consists of one single N-terminal, that is amino terminal, and one C-terminal, which is a carboxy terminal. So if I have any protein where I have only one N-terminal and one C-terminal, we refer to it as a single peptide chain. And when I have single peptide chain, and these peptide chain, they can go to another level of structural organization where each polypeptide chain is referred to subunits. Now we are going to talk about interaction between subunits. That means we are going to talk about tertiary structure interaction. Now, one concept which I would like to clarify at this point of time is that for a given protein structure, if I say primary structure of a protein, now this primary structure of protein defines the amino acid sequence, but it does not give you a functional protein. It has all the amino acid, but it is not in the natural form or a native form, and that's why they are not active. If I refer to the secondary structure, it is a complex structure. It is a spatial arrangement where proteins are interacting with a neighboring amino acid, giving you a structure, a spatial arrangement, but this is not the functional structure. So what is the functional structure of the protein? So minimum functional structure of a protein is when they attain a tertiary structure. So once tertiary structure is formed, they can be functional. In most of the protein, it is possible that tertiary structure is a functional. But in certain proteins, their native structure is as such where more than one tertiary structure interacts with each other to form a native structure. Do you understand what I mean by native structure? Native structure is a structure which is present in a natural form. So if I isolate a protein in their natural form, whether they exist as a tertiary structure or quaternary structure, that will be decided based on how they exist in nature. So any tertiary structure is a structure which is, you know, which is a potential to give you a functional structure. So minimum functional structure is obtained by tertiary structure because it is a folded, it is a spatial arrangement in 3 dimension, and it takes care of all the interaction within a single peptide chain. So any peptide chain which is having a single peptide, it will maximum attain up to tertiary structure. But there may be protein which may have more than one tertiary structure, and they are the one where you have a quaternary structure, another level of organization. And here, each tertiary structure, each protein part where tertiary structure has been attained is referred to subunit. So what it means is that in a quaternary structure, you will have more than one tertiary structure which are interacting with each other. So how is this, organization? In a primary structure, you have, amino acid sequence. Secondary structure, you have a primary structure which has a folded by neighboring interaction. In tertiary structure, there are many secondary structure which are interacting with each other. So locally there is a folding, and this local folding interacts with each other to give you a tertiary structure. And once you have a tertiary structure, there is a possibility. It is one possibility is that they're fully functional. Another possibility is that these tertiary structure interact with each other and give you another level of structure, which is referred as a quaternary structure. So let us see what are the quaternary structure. So first one, on the right hand side, you see a protein which is present in bacteriophage, lambda bacteriophage or viruses. It is a dimer. You can see that there is two identical unit. Now, on one side you have magenta, other side you have yellow color. So the spatial arrangement of subunit and nature of their interaction. So how these two subunits they are arranged and how they interact with each other is referred as a quaternary structure. This is one of the example which I am taking is a dimer, means two proteins, two tertiary structure interacting with each other. So they may be similar structure, they may be different structure. The simplest one I'm taking about is where two identical subunit, two tertiary structure. When I'm talking about subunit means a complete tertiary structure. It itself has a secondary structure, it has a folding, it has all the interaction. Now it interacts with another similar structure. So two subunits having identical structure, they interact. So these are called as a dimers. So example of this is DNA protein CRO found in bacterial virus called as a lambda. So it is a lambda phase where you get this kind of interaction. The more complicated structure or quaternary structure, where more than one type of subunit can be present, often in variable numbers. So what it means is that one simplest structure could be that in a quaternary structure, two tertiary structure or two subunits having similar structure, they interact with each other. So we call as a two subunits and they are having similar structures. Now it is also possible that there are different types of structure at the tertiary level and they start interacting. So the example is given on the right hand side, bottom side. Let us analyze it. So it is a slightly complicated quaternary structure where more than one type of subunit can be present. So each subunit will have its own structure. Each subunit refers to a tertiary structure of a protein. So what it means? It means that if I have a quaternary structure, it will have subunits. Each subunit independently will have complete N-terminal, C-terminal, all of them, they will be arranged in their tertiary structure, and each tertiary structure will interact with each other. So here, the example has been taken as a human hemoglobin. It is oxygen carrying protein in blood. and consists of two subunits. When I say subunits, what it means? It means that each subunit will have different structure. So each one, if I analyze, their tertiary structure will be different. So this protein has a two subunits of alpha and two subunits of beta. So how many subunits? Four subunits. And in this four subunit, two subunits are similar and two other subunits are similar. So two of them which are similar we call as the alpha subunit, and two which are slightly different, they are called as the beta subunit. Now, a small change in the arrangement of subunit within the hemoglobin molecule, they will allow it to carry oxygen from the lungs to the tissue with a great efficiency. This is a very interesting thing. How these subunits, they arrange within themselves, that gives you a possibility that how they are going to work. And that's why probably the quaternary structure has been aligned. The structure of two identical alpha subunit, they are given in the red. So on the right hand side, if you see, there is a reddish structure on the top and the bottom. You can see one of them is one alpha subunit, another one is another alpha subunit. These two alpha subunit, they will be similar. Then you see two of them in yellow color, and they are the similar structure. So they are a beta subunit. Other than that, you are looking at the blackish structure. This black structure refers to the heme. Now, heme groups, they are arranged with this. So there are four subunits, two are similar, another two are similar, and that is where we call it as a tetramer. So two alpha, two beta subunit giving a tetrameric structure. Now, can we have more than four subunits? Answer is yes. Now, you look at the viruses, which make most of the limited amount of genetic information, and this is done by forming codes, and that use the same kind of subunit. Many times they are repeated, and they are making a symmetrical array. One of the virus, which is known as a rhinovirus, it causes common colds. It's a very common. It has a 60 copies of each four subunits. So you have four subunits, and not only one such subunit, one such code protein or one such protein is present. There are 60 copies of such subunits are present. So subunit comes together to form nearly spherical shell. So you look at on the left hand side where you have a green, blue, red. So these colors are present, and what you have is that they form a repeated structure. So there are 60 copies of this structure, and what the combination gives you a kind of a spherical shell. On the right hand side, it is given an electron micrograph showing a rhinovirus structure, and it looks like a particle, but actually each one will have 60 copies of this repeated quaternary structure. So this gives you the possibility that how the proteins can be organized. So we through the amino acid organization, we have talked about primary, secondary, tertiary, and quaternary structure. Now let us see how we determine that what kind of structure gives you a function. So here, very fundamental experiment was done to demonstrate that the amino acid sequence of a protein is the one which ultimately decides what should be the structure. So if there is a particular set of amino acid, they will tell you that this type of structure is likely to be formed. Let me again re-emphasize that by just knowing the primary structure, you can predict a tertiary structure, but you cannot be 100% sure. And I'll tell you why it happens. Number one. Number two, some of you may ask that how do we know that actually it has a spatial arrangement? Why not that a single straight chain protein is giving us a function? So these questions will be answered when we show you this kind of experiment. So there is a classical experiment done by Christian F. And what he did almost in 1950s, around 70 years back, what he did is that he has taken an enzyme which is known as a ribonuclease. And so ribonuclease is a protein. It has given you a tertiary structure. And what he did is that, so just to define, it is a single polypeptide chain. So tell me what structure it can gain. If I have a ribonuclease which has a single polypeptide chain, can it go to quaternary structure? Yes or no? Can it form quaternary structure? Any one of you? Any answer from your side? Let me tell you that the primary thing which is important for giving you a tertiary structure is that they should have more than one peptide chain. So if I have a single peptide chain consisting of 124 amino acid residue, of course, they have a cross-link by 4 disulfide bond. That means it will not go to the quaternary structure. It will remain, it will go highest level of structure will be tertiary structure. So what you are looking at is there is a one amino terminal and there is a one C terminal. Can you see this? I'm just highlighting that. So you have one amino terminal, one C terminal, and what has happened that these are the amino acid sequence. So you are learning KETA. So these are single residue coding for amino acid. Now this amino acid has formed a turn, and now here what you see is that here you have a CC bond. So you have a, what is this? Cysteine interacting with each other. These are disulfide bond. Here again you have a disulfide bond. Here again you have a disulfide bond. So this disulfide bond, when they interact within the protein structure, they give them a tertiary structure shape. So in a bovine ribonuclease, what you have learned is that there are four such interaction. Now these interactions, they lead to, so where is the fourth one? There is the one which is, I'm just trying to make it. So this is the fourth one, which is hiding here. So these are the sequence 1, 2, 3, and 4. These four CC bond, which is nothing but cysteine, making cysteine, and these are disulfide bond. So a very simple structure. How to convert this structure into a straight chain? One thing could be that you just break these CC bonds. So what you can do is that you can break this disulfide bond. If I break this disulfide bond, they will not hold each other and they will make a random sequence. So what has been done is that he has destroyed the three-dimensional structure. What is three-dimensional structure? This is the structure where especially the protein is interacting, the amino acid residues, they are interacting with each other. So what he did is that he destroyed the three-dimensional structure of an enzyme, this is the ribonuclease, and then determined what conditions were required to restore the structure. So he started learning what is happening. When this enzyme was treated with beta-mercaptoethanol, so he has used beta-mercaptoethanol, 8 molar urea. So what will happen? Beta-mercaptoethanol, break the SS bond, then 8 molar urea will denature, and the product was fully reduced. So the ribonuclease, they were reduced and they formed randomly coiled peptide chain. Now, when he has disturbed the structure, tertiary structure, what he found is that there was no enzymatic activity. That means there was no function was retained by the enzyme. What does it mean? Any one of you? I need answer from you. So if I say there is no enzymatic function, that means once you break the tertiary structure, convert them into primary structure or random structure, which is basically primary structure, it will not retain its function. So the critical observation that was the denatured ribonuclease freed of urea. So now what he did is that he removed urea, he removed beta-mercaptoethanol, and when he did so, the enzymatic activity was regained. Of course, he was very lucky so that he could get experiment done in such a way that he could remove the enzymatic activity and he could regain the activity. In some protein, it may not be regained also. So this is a very robust kind of a molecule which gives you this structure. So on the right hand side, you see what is happening. He has used this beta-mercaptoethanol. He broke the SS bond and he denatured the protein, and what he could get is he could get a protein. This protein is not having a tertiary structure, and that is why it was not having functionality. So he reduced the disulfide bond. So what will happen to reduce disulfide bond? SS bond will be broken into SHSH. And now since they are not linked, so the force which is holding the protein tertiary structure will not be there. And that's why they will achieve a different kind of conformation. Now what he did is that he removed this beta mercaptophenol. And what he did is that he removed the urea, which was denaturing. So then what he observed is that catalytic activity of this was regained. So the general principle arrived from the Effinson experiment on ribonuclease was that there is the information which is present in the sequence, and that is what is important for giving a three-dimensional structure. Now let us move on to the similar experiment a little bit more. What he did is that he used he got a very different result when he reduced the ribonuclease, which was reoxidized while it was still 8 molar urea, and then he dialyzed to remove urea. So first thing is that he did oxidize. So what he did is that he used molar urea and beta mercaptophenol. This kind of a structure was formed. This experiment is a little tricky, so try to understand. See, this is the tertiary structure, native structure of ribonuclease. This is the structure where he has used 8 molar urea, beta mercaptophenol. The structure has opened, and this tertiary structure is lost. So we call them as a denatured structure. So you have a native ribonuclease, you have a denatured ribonuclease. Now what he did is that he reestablished the bond between the disulfide. Uh, so he has reestablished the disulfide bond when under what condition? He has done this while it was still in 8 molar urea. When 8 molar urea was present, he tried to establish this disulfide bond, and then he removed the urea. So what it means is that there were some disulfide bond which were formed were not the original native protein. So what ribonuclease oxidized in this way had only 1% of enzymatic activity. That means the structure which was formed was not the native structure. It was slightly different structure. So what it means is that there are different ways in which pairing of these 8 cysteine residue molecule can occur. So what you have seen is that, see, there is one pair, there is another pair, there is another pair. So there are 4 pairs. So 8 cysteine residues are there. They have different ways in which they can recombine. So now, if I say that this residue is with this residue, so the story will be different. The protein will not gain its original structure, its native structure. And what will happen? Then it will not regain its function. So very important thing is that when the protein is denatured, and after that renatured, during the process of renaturing, they should attain their native structure. Otherwise, there is a huge possibility that they will lose their function. So for functionality, it is important that they should gain their native structure. Now we are looking at the residues. Now question comes that in this we have shown, in the previous slide, we have shown that how the recombination occurs. Now there is one more experiment is possible, that there are 105 ways and there is only one way of correct pairing. That means there are 104 ways in which the incorrect pairing can occur. There are 104 ways in which they can have incorrect way of interacting with each other. It's interacting with say instead of interacting with 40. Been shown ribonuclease structure will not be functional, or if it is a functional, its functionality will be very poor. The scrambled ribonuclease convert into fully active native ribonuclease with a trace amount of beta mercaptophenol. Now, the added beta mercaptophenol, what it did is that it catalyzed the rearrangement of disulfide pairing until the native structure was regained about in 10 hours. So there are different ways in which reorganization was done, and the only way by which you can regain its native conformation gave the functionality of the protein. So what is the conclusion from these slides? Hello? Is anybody present?
Yes, sir.
Yes, sir.
Yes, sir.
Okay, tell me what you have learned from these slides. You have learned where very basic principle is that as long as correct confirmation or native confirmation is achieved, you will get the functionality. If the native structure is not achieved, you will not get the function. So it's very, very important that you regain the native structure. Now moving on to the another part, that so this has very clearly demonstrated the structure, how correct structure is important in the protein functionality. You have learned about the quaternary structure. Let us know that how we can say that a particular structure will be formed, where this message is given. So amino acids have different propensity of forming alpha helix, beta sheet, and beta turn. This is a very fundamental thing, and this gives you all the knowledge about the proteomics, or something where we talk about the bioinformatics of protein. How we learn about the bioinformatics, how we get the information. So suppose I give you the primary structure, can you tell something about the tertiary structure or quaternary structure protein? Answer is yes. So I'll talk about a little bit about this, and to learn this you have to understand a slight points. And as you have learned in case of the previous experiment, that it is not possible that even if we have the same amino acid composition, the tertiary structure you get is going to be the same, because some other interaction. So it depends on the random interaction between the amino acid which can lead to the tertiary structure. But yes, there is a possibility. There is a possibility where you can predict the local interaction. So how do we do that? These amino acid, I'll give you a list of amino acid, not necessary that you have to cram them, but to understand them, that these amino acid have certain propensity to form different types of structure. So the different type of propensity will lead to tell you that these amino acid have more, chances to form alpha helix. These amino acid have more chances to have a beta sheet. These amino acid will have more chances to form beta turn. So residues like alanine, glutamate, leucine, they tend to form alpha helix, whereas a valine isoleucine tends to form beta strand. So this information is with you. So if I look at the structure, if there is a pattern of alanine, glutamate, leucine, I'll say that this is going to ultimately give you alpha helical structure. But it is to certain level of confidence, not with 100% confidence. Same way, glycine, asparagine, and proline have a propensity for giving you turns, or they are present in turns. The branching in beta carbon atom in valine, if you look at the valine residue, what you find is they have a beta carbon atom. Same with threonine, isoleucine, they tend to destabilize alpha helix because of the steric classes. So this is all theoretical thing, and of course that is what happens. So if you have valine, threonine, and isoleucine, they will be mostly present in the beta sheet conformation. And why it is so? Because of the presence of a beta carbon in these amino acid, which destabilizes alpha helix because of the steric hindrance or steric clashes. Readily accommodated beta strand in which their side chain project out of the plane containing the main chain. So what happens, uh, some of the amino acid they can so these amino acid valine, threonine, isoleucine, they can form beta strand. Why they can form beta strand? Because these can be accommodated in the beta strand, because they are in the side chain and they are projected outside the interaction, so they will not interact with each other. Same way, you will find the serine, aspartate, asparagine, they tend to disrupt alpha helix. So you have a set of amino acid which are alpha helix disruptors. And if they are disruptors, that means they should be present, and there is a very high chance that these amino acid are going to be present in the beta sheet conformation. So these side chain contain hydrogen bond donors or acceptor in close proximity in the main chain, where they compete the main chain NH and CO group. So there is a very interesting reason which is given for serine, aspartate, and asparagine. All of them, they have hydrogen donors. And if they have hydrogen donor, what they do is that they compete with the secondary structure formation, where NH CO group, they are present in the main chain. They are also forming the interaction. So you are having two types of interaction. One interaction of hydrogen bonding is because of NHCO group, and another is a hydrogen bonding because of the side chain. NHCO interaction is because of main chain, and serine aspartate, their side chain, they will interact. They will also give you another interaction. Why serine? Because it has OH group. Why aspartate? Because it has COH group. So their side chain has additional hydrogen donor, and that's why they will compete with the main chain interaction. Proline is another amino acid. What is unique in proline? Do you remember? Proline is the one where its side chain interacts with the main chain. So proline disrupts both alpha helix and beta sheet strand, and and it lacks the NH group, and because its ring structure restricts this F is 5 value, which is around 60 degree. It is not minus 60, it's a 60 degree. And that is the way you learn about these amino acids. The glycine readily fits into the structure, and you will find that there are many proteins where glycine is predominant. So this slide gives you a list. No, I don't want you to learn about them, but you must understand that they're amino acid, like these amino acids. They have more value for alpha helix. Sorry, let me just go to the previous slide. So here, what you have learned is that there's certain amino acid which has alpha helix value more than beta sheet. So what it means is that or turns. So they should be present mostly in alpha helix. Now there are some amino acid, for example, this amino acid. It has more value for beta sheet than alpha helix. So these amino acid will be mostly present in beta sheet. There is some which may have higher value for turn, so they will be most likely present in the turn. So if I give you this table, you should be able to predict that which amino acid should go in which type of structure. So the value given here gives you that there is a propensity of each amino acid to form a particular shape in the secondary structure. But it is not always true. A particular shape can attain different conformation. This tells you that is where the bioinformatics fails. Why it fails? Because some of the amino acid, same sequence, can have a propensity to form alpha helix, and it can also form beta sheet. So this is a kind of a prediction, and now I'll tell you how this prediction can become more useful. So that part we'll be talking about. Now let us talk about when I say protein folded and unfolded form. The folded form is the one which is a native form, the unfolded form which is a non-native structure or a denatured structure. So what you did is that, like if it's an experiment, a person, what he did is that he has taken a protein and then he started increasing the concentration of denaturing agent. So what is a denaturing agent? Like urea, like your the guanidinium hydrochloride. So you keep on increasing the concentration. So what has happened up to this concentration, that means at lower concentration, all the protein was folded. So you can see that the protein is folded up to certain concentration of denaturation. So this is the denaturant concentration up to which protein is folded. Now at 50%, this 50% concentration, what you find is that 50%, that means if I have a four units or four molecules, two of them will be unfolded and two of them are folded one. So you have 50% folded for 50% unfolded. But if I reach the protein unfolded, here you find that 100% is unfolded. So there is a time when you have 100% folded, 100% unfolded, and there is a concentration of denaturant where 50% is folded, 50% is unfolded. Now looking at the protein fold, so the question comes how protein folds. You have learned the protein can be unfolded, that means the denatured form, or it can be in the native form. Now there was a very interesting experiment was done and some theoretical value which was obtained, and it shows that the cooperative folding of protein, which is a thermodynamic property, it reveals nothing about the kinetics and mechanism of folding. So the folding, the way protein folds, it doesn't tell you about the kinetics, it doesn't tell you the mechanism of folding, how it was done. So Leventhal, he was a scientist. What he did is that he took 100 residue. He just took a consideration that if I have a protein which is made up of 100 residue, what are the possibility? How many types of interaction they can have between each amino acid, and that's the way they can form a structure. So what he found is that there are around 3,100 types of structure can be formed with this particular type of structure. Now, if which is equal to almost this many combination, and if I take around 10 to the power minus 13 second, where one conformation is converted into another conformation, the search time would be something like these many seconds, and he converted all those things, which comes out to be 1.6 into 20 to the power 27 years. That means it is not possible. So it clearly says that it takes too long for even a small protein to fold properly by randomly trying out all possible conformation. So this was called as the Leventhal paradox. Leventhal paradox is that enormous difference occurs between calculated and actual folding time. This is very interesting. That means if I go by theoretical value, considering that there is a random interaction between each amino acid, and that's the way the folding occurs, which is not possible. Now, what he did is that he also learned about the free energy difference between folded and unfolded protein. So a typical protein having 100 residue around have 10 kilocalorie per mole energy. So, and each amino acid will have 0.1 kilocalorie. If it is 100 amino acid, 1 kilocalorie. So energy required to keep them in a folded structure is very, very small. And this amount is less than the thermal energy. So when I talk about 1 kilocalorie per mole, recall that we have discussed around 0.6 kilocalorie per mole at room temperature, which makes the protein to be stable. So stabilizing protein is through different process. So we will be talking about that. Here, very interesting experiment was done. Look at this. Do you see this structure? And do you see this structure? So it is like a monkey sitting in a typewriter, looking at the random, structure, and he is just putting, his fingers without knowing the sentence, and he's writing Hamlet. And it's possible that they have done the computer simulation, and they found that what kind of, thing can emerge by random hitting the computer keys or a random hitting of the typewriter. So this is something gives you the idea that this is not the random interaction, there is some other way of interaction. The sequence of amino acid completely determines the three-dimensional structure of the protein. It is known. However, the prediction of 3D structure from sequence has proved to be extremely difficult. And otherwise, what can happen? People, they can just you give me the amino acid primary sequence, I will give you the final structure. It's extremely difficult. And that's why, where bioinformatics is stable up to some prediction level, but after that it fails. So local sequence appears to determine around 60 to 70% of the secondary structure. So if I look at the local interaction, as I said, that these amino acid will give you the alpha helical structure, these amino acid has a tendency to form beta sheet. So by that kind of a prediction, we can give 60 to 70% secondary structure. But how about 30% structure? We cannot fix. So what you require is you require long chain range interaction. That means interaction globally, how the amino acid interacts globally. So first, ab initio prediction. Ab initio means from scratch, from the basic amino acid sequence, which attempted to predict the folding of amino acid sequence without direct reference to known protein. So one way is that I take amino acid sequence and I start predicting what could be the third tertiary structure without looking any protein structure to compare with. So there was a computer-based calculation. They were employed to attempt to minimize the free energy structure and give an amino acid sequence to simulate the folding process. And then these methods were limited. So one way is that what I do is that I give the feed to the computer saying that these are the amino acid which is going to give you alpha helix. These are the amino acid which is giving you beta sheet. And that's the way we predict the folding in that three-dimensional structure. But that failed. That didn't work. But in this method, what they did is that there was many possibility. and the marginally stabilized protein and subtle energy of weak interaction in aqueous solution. So that's why this prediction was not foolproof. So another approach was that what you do is that you take a protein where three-dimensional structure has been determined. How we determine the three-dimensional structure? Experimentally, it is done by X-ray crystallography. Now, I have not given you the detail how primary, secondary, tertiary structure is determined, or quaternary structure is determined. The primary structure is determined by sequencing amino acid. So here, what we do is that from N-terminal, each amino acid is determined, and there are different, amino acid sequencer. They can determine you the amino acid sequence. And here, the basic thing is that we see the arrangement of amino acid in the straight chain without folding. The secondary structure can be given by 2 or 3 techniques, and these techniques are CD spectrometer. That means we have circularly polarized light which is passed through the solution of the protein, and that will determine that how the interaction occurs in the protein in the neighborhood. So CD polarimeter can give you the structure, or CD can give you the structure. CD is nothing but circular dichroism. The second technique which is used to give the secondary structure of protein is known as FTIR. That is a Fourier transform infrared spectroscopy. The tertiary structure of a protein is given by X-ray crystallography, and that's the give you the complete structure. So suppose I have a protein where I have determined the tertiary structure. I use that knowledge that which amino acid is giving you the a particular sequence is giving what kind of a structure. Based on that, if I give the structure, that gives a relatively more closer structure to the native structure. Interesting thing is that protein modification in cleavage confirms new capabilities. So if I take a protein and modify or cleave it, it gives you new kind of a function. And there are different 20 amino acids, and these amino acids, they can be covalently modified. And what you can do is that sometimes they can also improve the function. So there is a very common thing which has been done in protein, and that is done by looking at the groups. So suppose I modify the amino terminal by acetylation. So this has been done, and this modification of this protein gives resistance to degradation. So protein, they are very, not stable, and unstable structure generally degraded. So if I modify the protein terminus by acetylation, that helps in the making them resistant. Same way, there are hydroxyl group. Where are hydroxyl group? The hydroxyl group to many proline residues stabilize fiber of newly synthesized collagen. So I will later on tell you that the collagen fibers, they are stabilized because of the presence of hydroxyl group. So if proline is converted into hydroxyproline, then you will find that your protein is good. Otherwise, protein will not be stable. Then in vitamin K deficiency, insufficient carboxylation of glutamate is leading to the hemorrhage. That means clotting is not proper. So glutamate, if it is carboxylated properly, the clotting occurs properly. So there are these functions which are influenced by the modification of certain groups, which I have talked about. Many proteins which are especially present in surface of the cell are secreted. What they acquire? They acquire a specific asparagine residue. So this kind of a modification, so protein which are secreted protein which are present on the surface, they acquire asparagine. The addition of sugar makes the protein more hydrophilic. That part we have discussed talking about the carbohydrate. So if we have a protein, I modify it with a carbohydrate, that will give you more, more hydrophilicity. And same way, if I add fatty acid in an alpha amino acid, the cysteine sulfhydryl group produce more hydrophobic protein. So you can take a protein, modify them, give them a more hydrophilic or hydrophobic structure. There are many hormones. I'll quickly, in 1 minute, will try to finish it. They have many hormones like epinephrine. Their activity is altered by phosphorylation. There are many proteins, they trigger their phosphorylation of hydroxyl group of tyrosine residue. Amino acid. So when I say tyrosine residue. This is one of the amino acid which will be modified. So there are phosphorylation, dephosphorylation process. And last very interesting example is green fluorescent protein, where what happens that you have a set of amino acid which is tyrosine, serine, tyrosine, glycine, and here this set of tripeptide, they are arranged and their oxidation leads to the very interesting phenomenon which is the fluorescence. So this is the protein, this is modified during in presence of oxygen, and you get the fluorescent structure. So the structure of green fluorescent protein, the rearrangement of oxidation of serine, tyrosine, and glycine in the source is the source of fluorescence. So once these proteins they are oxidized, there is an arrangement, and that gives you a fluorescent behavior. So I'll stop at this point. Any questions? If there is any question, we'll address. Otherwise, if you have any question so far, write it down right now, and in the next class, you can ask the question. I'll address. You have two modes of asking question. One of them could be that you can ask question by writing your comments, or you can write mail. Or mail will be slightly difficult. I would like to address all these questions here itself whenever you are in class. Okay, then I'm going to sign off. Thank you very much.